So, hello everyone. Yeah, very excited to tell you a little bit about my work. So the idea here is because this is the first talk in this series, the spring series. I'm going to spend a little bit of time in the beginning talking about the principles behind lighting in health and physiological effects of light, before going into three different projects of mine. And for all of them, the idea is to obviously show you the study and the results, but also connect those dots a little bit to how this might inform lighting design and the application of the whole topic. So before we start, I think it's important to tell you where I'm coming from. So from which lens I'm looking at the topic. And actually, I'm coming from very far. Started out as an architect, basically, lighting designer, and at some point I got involved in a project where it was all about defining healthy light for people. And I got so interested in the topic that it kind of stuck to me. And all of my professional and academic life is now dedicated to that topic. I did my PhD in human biology following that. And so now I, hopefully help to bridge the gap between those two topics of basic scientific research and the application in the field. So, yeah, let's jump right in and let's make that connection why physiology and light are even connected. So the topic I'm going to talk about obviously has much to do with rhythms. So let's start there. Hopefully all of you are well rested. So I'm up for a few hours. Anna just got up because she's in a different time zone right now. But if we look at our body and in internal rhythms, what we find is we don't work the same way at all times of days. So there is a rhythmicity to it, and this rhythmicity really is very much it's very different for different parameters. You're looking at core body temperature or cortisol, but mostly all parameters of the physiology have some kind of rhythmic behavior. And this whole orchestra really ends up affecting us in a way that we can feel. So our mood or our alertness changes over time, and in a very rhythmic manner. And so yeah, we can recognize that this is happening. But if we look back into more of an evolutionary aspect, we also see that our environment is rhythmically changing very much. And together with the environment, and there is a causal relationship there, the lighting environment also changes very rhythmically. So during daytime, we had and continue to have daylight available, which is a very specific characteristic spectrum. And during the night, we don't have any kind of light at all. Well, right now we do have, but not from an evolutionary perspective. And in the evening, during most of the time, there wasn't any light available as well. But in more recent evolutionary aspects, there is firelight. But it's not only specific in terms of the light spectrum, but also in terms of the distribution of light. So daylight coming from a large source above of us, and firelight coming from kind of a small narrow point source in the lower visual field. And because of these stark changes in the environment, it's not surprising that evolution has found some way to connect these things. So what we found is that in addition to the photoreceptors that we use mainly for vision, the rods and cones, and here is our sensitivity to brightness shown here. We also found, and you could debate newly discovered, because we know about these receptors for about 20-25 years now, that there's a third kind of photoreceptor that really acts like a daylight sensor for our eyes. So it has like very low resolution, is sensitive in the more bluish range of the spectral wavelengths. And this sensor communicates the dynamic changes of light and darkness into our brain, like takes a completely different route from visual perception straight into our autonomous nervous system, where we do have the master clock that relays this information to every cell in your body, and that affects how we work on an organ level and on a system level as well. So that's basically where these rhythms come from in a large part. From the collected knowledge that we have following like this discovery of the pathway, recommendations for healthy light exposure for daytime and nighttime were developed. So there is like this huge consensus view paper from Brown and colleagues that is very widely known in like the people familiar with the matter in terms of the lighting design, but it's also like a huge step for the science community to have been able to come together, look at the available data and say, well, those are good exposure values that people should get in order to get at least some minimal dynamic range. And if we condense that information, it really comes down to like a 3-step process. During the daytime, you should reach at least 250 lux of daylight. And 2 to 3 hours before sleep, you should reduce that to less than 10 lux. And during the nighttime, during sleep, you should have no light at all, but keep it below 1 lux. And those are daylight values, but we obviously are exposed to other types of light, other spectra. And so there is like this CIE standard 026 from 2018 that allows us to convert basically every spectrum to a measure for the non-visual potency of light to the melanopic system, which is then called melanopic EDI, or equivalent daylight illuminance. So like with this kind of dynamic, this is a recommendation that people should follow. What we find, if we look at the light exposure people really have out in the field, it's, they are not getting that. So here is like one example day of a person's light exposure measured very near to the eye. And here are these recommendations I just showed you. And every time we do have like a gray dot here, people were, or this person was outside the recommended values. And you can see it's like most of the daytime. So the general vibe is we have not enough light during the day and too much light at night and in the evening. And this has like negative health effects. So just to very quickly go into 3 studies that, take that message home. There's a study from Rob Lucas and Altuk from Turkey, looking at a kind of a smallish sample of UK adults. And they also found that those recommendations are usually not met during day and night time, and that there are some correlations between sleep timing or sleep onset and sleepiness in the morning, depending on how much light stimulus people had. But this goes beyond sleep, looking at another study from Kim and colleagues, also looking at light exposure during the night. They found that there was a considerably higher prevalence for metabolic issues like obesity, type 2 diabetes, hypertension in people who had like more light during the nighttime. And another study, this time really looking like kind of at a big data set, over 80,000 people, which was very interesting because it looked at mental health problems associated to light exposure, and it found you increase the risk, or there is increased risk to have certain mental health problems if you have more light during the night, but you reduce the risk by actually having less light during the day. So there is like this dynamic of day and night light exposure encapsulated in those results. These are just some examples of where all this knowledge can end up if you translate the topic into a technical solution. If you want to control how light exposure, affects people. There are several alleyways. One is, to give them a certain lighting design. The other is to make them aware of that and change their behavior. And because, I'm coming from a lighting design background, this is the kind of area where I have influence and the possibility to do stuff. So like these are projects where all of this knowledge ended up in, forming the way the lighting system is set out to function. And while we are not looking at those projects in itself, what I want to do is look at certain research projects I did that in some way connect to, how a lighting design, might be approached. And these three projects don't have any kind of overarching narrative. So this is more about showing you like the width or the breadth of projects that are possible in this field just from like one person alone. And I'm certainly not covering all the bases. There are many more diverse studies in that field. But yeah, these are three examples like that show how far things can go and are relevant for the kind of lighting we make. Let's start with the first one, which is all about pupilometry. And, to get you into the vibe, here are some pupils. So we looked at a lot of people or at a lot of pupils, I would say. And the pupil is basically controlled by two muscles. So you have like a radial muscle and you have a sphincter muscle, a circular muscle. And those muscles are innervated by two different parts of your autonomous nervous system, by the sympathetic nervous system, which widens the diameter, and the parasympathetic system, which constricts the pupil. And the balance between those two systems and consequently those two muscles set the resulting diameter. And this system really gets input from all the photoreceptors in order to set the diameter. So it has other inputs as well. So not just like photoreceptor input, but higher cortical influences, for example. But in general, it's controlled by the photoreceptors. And what was found in the, I would say 15 to 10 years ago is that the IPRGCs that are responsible for all the other circadian effects that we briefly touched upon have an effect on the pupil as well, and especially on the long-term diameter. So if you stimulate the pupil with light, it will obviously constrict. And then if the light stimulus is it's offset, then the pupil redilates to its initial diameter. But if you have like a strong stimulus to the IPRGC, then like the redilation time is certainly prolonged. And if there is some kind of lesion with the IPRGCs, there really is no difference. So if you have like a red stimulus, which doesn't stimulate the IPRGCs very much, you get a strong redilation. And if you have like a blue stimulus, which is very intense for the IPRGCs, then you should see like a different pupil behavior. And like this is very interesting for lighting design also, because it's a diagnostic parameter. So you can use it as a diagnostic instrument to see whether it would even make sense for a certain population to do a lighting intervention. So if you're working with older age people who might have already some lesion in the IPRGCs or in the pathway to the brain, you might have to use a pharmacological route in order to bring them better sleep, for example. So this really would be like a great diagnostic instrument to see how people are doing in terms of the non-visual stimulus. What would be even greater is if we could just record people's pupil and see how much their non-visual pathway is affected by the light that they are exposed to right now. And that means continuous light exposure. So here we have like a very short stimulus, a few seconds, and then you have like this redilation phase. That's not a normal circumstance. So what we wanted to do was look at a more prolonged stimulus and see what kind of wavelength sensitivity we would get. And here's the basic experimental setup there. We have a light dome that we can set to different narrow band lights. We have a pupilometer where you can record pupil diameter, and then we have a program that creates a certain stimulus series. And so we started out with 2 series, like one was called up and one was down, and you can see it's a sweep through the spectrum, and each spectral phase lasted for 15 seconds. And so based on what we found in literature, we expected that about after 10 to 15 seconds after stimulus changes, only the IPRGCs should be like the main contributors to the pupil dilation. And we measured this by starting out with dark adapted pupil. That was our baseline. And then we looked at the pupil constriction, normalized pupil constriction at certain points in the stimulus series. And so, for example, here, this would relate to an 8% NPC, this to an 11% NPC, and this to a 13% NPC, and so on. And so this was our dependent measure. And because we had like many influencing factors, like not only the wavelength, but also the sequence of lights, what kind of chronotype people had, the time of day, and so on. We used like a combined parametric, non-parametric method, which uses generalized additive mixed models that are able to accommodate all these different parameters, and also use like a spline-based method to have non-linear relationships modeled. So we looked at that for a number of people, like 75 people went through that, and what we found was actually like very, very disappointing. So what we found was this relationship between the normalized constriction and the wavelength that you can see as a spectrum here. And I've printed the action spectra for the IPRGCs, for the rods, and the brightness detection right here. And what you can see is basically if you do these 2 sweeps and from that analyze, what kind of behavior do you have for wavelength sensitivity, we don't see IPRGC dependency. So we were asking ourselves, why is that? Why don't we see that? And one of the questions might be obviously, well, I mean, you're doing this smooth sequence through the wavelengths. Maybe that's a factor in that. And so we did a second experiment based on that. So we went back to the drawing board and we said, okay, let's mix up like the order in which spectra are presented. But because for technical reasons, we couldn't switch that immediately, because like there is a rod in there, a monochromator rod that has to move to that section. We added like those shutter periods where the shutter was closed. Meaning like there was a certain period of darkness in there. We wanted this to be as short as possible. It was 9 seconds at the longest. So we added like those 9-second intervals. And we also wanted to know whether like those darkness periods had any influence. So we added like a controlling sequence in there as well. And what we found was actually very interesting in that the sequence of stimuli presentation didn't have any effect on that. So if you look at the red curve here, we still see a like brightness detection behavior in there. But what did have like a very, very stark influence on that is if we added like those dark periods in between stimuli. And as you can see here, we have like a shift towards shorter wavelength in this scenario where we, in these scenarios where we added those dark periods in between. So this was very interesting to us because we didn't expect that. And when we looked at the data further, we saw that this is like a continuous process. So if we don't look at just the last 5 seconds of the stimulus, but like how this continually develops. So we have like 15 seconds here, and we have the wavelength on the x-axis again. What we are seeing is if we don't have, if we have dark periods between those stimuli, the center of, so the main sensitivity curve doesn't change its shape. So it stays centered on the brightness detection. But as soon as you, if you don't have any dark periods, so this is the same as the result we got in experiment 1. But as soon as you add those dark periods, we see a continuous shift in wavelength sensitivity towards shorter wavelengths. And so we went out of this experiment thinking, well, if the stimulus was like longer, would we then see an IPRGC dependency? So a stronger shift towards those shorter wavelengths. And as you might guess, like there was a third experiment building on that, where we looked at shorter, but also mostly here longer periods, where we had 30 seconds of light, 9 seconds of darkness. And indeed, like if we continue like to prolong this this sequence, what we are seeing is an even more pronounced shift towards an IPRGC dependent behavior. So the longer the stimulus lasts, if you have dark periods in between stimuli, you do find IPRGC consistent influence. But if you have continuous stimulation, which is the case like in every real life scenario, so not talking about the narrow band stimuli, but for longer periods, it's not a very good marker for IPRGC stimulation. So not what we wanted to have, but the results that we got, and we have to work with them. Another finding that we had, but this was purely exploratory, we didn't look for that specifically, but still, was that depending on whether you were more of an owl type or more of a lark chronotype, there was an interaction effect on your wavelength sensitivity. So the chronotypes were most sensitive to blue light in terms of their pupil diameter at the time of their preference. So like the larks during before midday, and the owls in the evening, which was very interesting because this is something that we didn't find in the published literature before, but there are some retinal mechanisms that might explain that. It's very interesting, but only an exploratory result. So something for another time to be replicated, hopefully. This was the most basic research I will talk about today. The next one is much more applied, but still looks at very basic principles. The question here was whether in terms of realistic lighting situations, does the composition of light in your environment, does it change an effort parameter? So you're doing a cognitive demanding task, and does this actually affect the effort? So does it matter basically how you light your workplace environment with 500 lux at the desk surface? And like this is a fake photo that was made after the fact, but it gives you a nice introduction in there. The background to the study was another study that was published by Lasauskite and Kajochen in 2018. And what they found based on correlated color temperature was that the more blue was in the spectrum, the less effort people needed to perform a cognitive task. This was indexed by the delta PEP parameter, which we'll get to in a moment. But for right now, let's just say that's all about effort. This was measured with impedance cardiography. And what you're seeing here is like a person that's getting measured lying down. So impedance cardiography is basically an extension to electrocardiography, but you're measuring impedance. And what's changing in impedance in your chest area is basically the amount of blood that you have in there when your heart is pumping it through there. And so in combination of electrocardiography and impedance cardiography, you can measure and calculate some cardiac parameters that are important for sympathetic activation, so for your readiness to perform. And this is like very sensitive compared to other measures of alerting effects. So what we did was we designed a lighting system, and we made three lighting scenarios that were very different in terms of the melanopic stimulus. Because what we wanted to know was whether what we saw from the Lasauskite and Kajochen paper was based on IPRGC influence in there. So as you can see, we have three stimulus levels at the observer: 241 lux, 182 lux, and 54 lux, and all of these are melanopic EDI, so the stimulus to the non-visual system. And we also have like differences in terms of how light is distributed. The fact that is kept constant across all scenarios is the light on the task. So we have 500 lux at the laptop at all task scenarios. So this referenced a high stimulus, a typical stimulus, and a low stimulus. And what we expected was that the stronger the melanopic stimulus is, the less effort in terms of delta PEP was needed. And we expected people to require more effort in the afternoon, and that the melanopic stimulus would interact with the time of day. We measured 27 participants in a within subject design at 2 times of day. And basically we had like a lead-in scenario, which was the low stimulus scenario. And then we had all 3 scenarios for every time of the day in random order. And it always consisted of the same order, which was like a baseline measurement of 10 minutes. Then taxing task was performed. And then we had a questionnaire afterwards about how people felt while doing it and how they liked the lighting scenarios. And now we get to this delta PEP part. Basically what happens is PEP is measured and calculated during a baseline and during the task, and PEP stands for the left ventricular cardiac pre-ejection period. So it's the period of isovolumetric pressure increase just before blood is ejected through the aortic valve. And the speed of like this isovolumetric pressure increase is only influenced by the sympathetic systems if you rule out some other conditions that might have an influence on that. And that's very different from other heart parameters like heart rate, for example, which is like very much controlled by sympathetic and parasympathetic influences. So lots of different systems pulling on that, and you have the balance in the end. For the PEP, you don't have that effect. You only have sympathetic influence. And if you calculate the difference between the task PEP and the baseline PEP, then you get this delta PEP, which is cardiac reaction or reactivity, and is an index for the effort a person had doing that task. The result again was like very surprising for us, because we did find an effect, but it was not like a linear effect, but rather a U-shaped connection between IPRGC stimulus and effort. So we saw that scene 2 led to the highest effort, which you can see as it is lower on this y-axis, and scene 1 led to the lowest effort, but not significantly different from the low melanopic stimulus. And in the late afternoon, we saw like this general change that we required more effort. People were up longer. This is like obviously what we are expecting, but we find that there is the same dependency between the different lighting scenarios. All of that, so these are changes in cardiac parameters. We didn't find any differences in performance or subjective alertness, task difficulty. So people performed basically the same way across all 3 lighting situations, but the objectively measured effort was higher in the scenario 2, which is like the middle melanopic stimulus. So obviously, again, this like was not what we expected based on our hypothesis, which is what you can see here in the green line. This is what we expected. We also didn't find this interaction effect between the time of day. But what this basically means is that there is very likely a different receptor type influencing that. So this is not singularly IPRGC based, but very likely cone influences are in there as well. And while this was not what we expected, it still is a very interesting result. And that is because if you look at it from a bit further away, like scenario 2 is the scenario that's modeled most closely to what typical office environments are in terms of the correlated color temperature, in terms of distribution of light, in terms of the light stimulus. And this one was the one that was least ergonomic in there because you needed more energy, which is basically what this delta PEP value breaks down to get to the same results. And 2 milliseconds doesn't seem like much, but it's something similar that we find in literature when, for example, a task gets more difficult, when we pay the people less money. I think like the difference in that study was 10 or so Schweizer Franken versus 1, or if the participant is in a worse mood. So these are like real effects that we also see by changing lighting design. Not dramatically, because all of these, remember, break down to 500 lux at the task level, but still, it is like a real effect, and it's about 5 to 10% of what the physiological range of the heart in terms of delta PEP changes can do. So it's a small effect, but I think it accumulates over time throughout a working life. So I think it is a good reason in order to provide different kind of lighting scenarios on top of what typical office lighting has. This brings me to the last project, and this one is much more application-centric. It's a project that actually started out as a pet project, because looking at the recommendations that I showed you in the very beginning, where we had 250 lux daylight during the day and so on, 10 lux in the evening, 1 lux at night. If we look at the studies that these recommendations are based on, the light coming to the eye is very much coming from a Ganzfeld stimulation. So like the whole visual field is in light, or we have a central stimulus. And what this means is light is coming in from the front. And this really is not the way light is distributed in our built environment. Light or high luminance light sources are at the fringes of our vision. And so I think a very valid question is, how does this relate to the amount of light you're actually receiving in real situations? So if we measure vertical illuminance, for example, to say, oh yeah, I reached my 250 lux, what you're actually measuring is a full hemisphere in front of a sensor. This would, for example, relate to a situation like this. So we have, this is a fisheye photograph, 180 degree. And you can see there are some luminaires in there. You have the light distribution. And if you overlay that with your binocular visual field for indoor environments, which is the colored space in there, you can see there are some of the luminaires that are outside this visual field. And you can already see the oversaturation here from the spotlights in here. So if you were to look up at them, they would certainly introduce glare. But it's not a problem for you. You don't even see them. But they would be part of a measurement, of a hemispheric measurement. So you would be doing you wouldn't introduce an error if you measured that as part of the amount of light that actually came to the eye. This is the same situation, but with two different lighting scenarios. And if we again overlay the binocular visual field, we can see this light source here is part of the visual field, whereas this one isn't. So even though both of these situations might have the same vertical illuminance, they have very much different stimuli to the eye. And if you measured that as part of an experiment, for example, or as part of lighting design, you would introduce some variability in terms of the effect of light, because what reaches the eye is actually the thing that matters. The CIE actually mentions or addresses this. So in the standard 026 from 2018, there is an annex where they recommend radiance hoods and cutting off the visual field at a certain degree, top and bottom. I think it's like 50 degrees to the top and 70 degrees to the bottom. And this suggestion changes if you're outside, because outside you have very much higher luminance levels. So you have a much smaller opening. But for indoor environments and typical indoor luminance values, this is like a sensible field of view. And so what we did was we calculated the field of view that we would expect according to the CIE and modeled that to the sensor opening of a spectroradiometer, which we are using to measure the spectral distribution and intensity for a given observer in a scenario. And you can see like this radiance hood here, this attaches to a spectroradiometer. And what we did was we made two measurements for a number of scenarios. So we were looking at, this is just a fisheye photograph of a sample scenario. We were doing like a 2π measurement, so a measurement without the measurement hood or the field of view occlusion, and then a second one with the field of view occlusion. And this obviously leads to two different measurements. One where you have the intensity and distribution of the full hemispheric field, and one where you just get the light that can enter the eye inside those angles. And here, what we have here is like the ratio between those two across the spectrum. And what you can see here in the end really is just noise, because like the intensity is so small that you get just a lot of measurement noise in there. And if you do the ratio of both of them, things get weird. But for the larger part of the spectrum, you can see whether the distribution changes between the spectral distribution of the full hemisphere and the field of view occlusion. And based on those spectral distributions, we did some calculations based on illuminance, based on melanopic EDI, and the daylight efficacy ratio. So basically the ratio between those two values. And like the most important one probably is the one at the bottom here, because this really tells you the impact of field of view occlusion. So how much light in this particular scenario is outside the field of view of a given reference observer that has like the field of view plus 50 minus 70 degrees. And you can see here it's minus 43%. So a considerable amount of light in this situation is outside the field of view. We can see it's similar to illuminance here. And so the spectral distribution between those two measurements didn't change by a whole lot. And this is something you can see visually here, but here it's like condensed into one number. And if we look at this number across a whole lot of different projects and a whole lot of different lighting scenarios, what we find is, so those are the ones, what we find is there is like a huge variance. And here we have like this Ganzfeld stimulation. So this is basically where all the studies that are responsible for generating the recommendations we have today lie in. They have a very narrow distribution. Most of the light coming in from the light sources there are validly inside the field of view. This is not the case for other situations. And we won't go into detail of all of them. You can look at the publication if you would like to, but I just want to address that there is like this huge variance. So there are some lighting situations where almost all of the light in them is coming from within the field of view. And then there are other situations, and this, for example, is a home office space where you have this luminaire above the workplace, and it's just cut off. It is not part of your visual field. And so no matter what kind of light distribution you might have, directly or indirectly, it really doesn't change how much light that gets occluded. And you only get just above 40% of the light that you would be measuring with a vertical illuminance measurement. So this thing really is a factor that we have to take into account. Very briefly coming back to the learning space that was part of the other study, because you already know it, what we can see here very interestingly is that the amount of light that is outside the visual field compared to inside changes between those three scenarios quite considerably. And it is actually helping our way of changing spectrum, because during the morning scenario that we dubbed the high intensity stimulus scenario before, we only lose 11% of light that is outside, because like those luminaires have a very large light distribution in our field of view. And as we get to the evening scenario, where less and less light is coming from these pendle lights and more light is coming from spotlights on the ceiling, the spotlight straight above you is really not part of your visual field, but it is part of the measurement environment of your sensor. So here a lot of light gets cut off, but that's actually something that we like. We want to have that. We want to reduce the stimulus to the eye in the evening or at night as much as possible. So if we were to take the ratio between the whole hemisphere of the morning and the evening scenario, we get like a much smaller dynamic than we get between the field of view occluded ratio between the morning and the evening scenario. And so if used smartly, we can incorporate the idea of the visual field occlusion into lighting design in order to make the dynamic range between day and night even more pronounced. So the implications I already told you about, limitations you can find in the paper as well. So I mean, this really was like a fast ride through three projects, but like overall, what I hope you to take away is that this is a very broad field that goes into many different directions, and overall just has so many different pieces where we have knowledge gaps and can look into, but yet taking one step back and looking at the whole field, it's so very important that we do this transition from science into lighting design because we do have those connections between lighting and health. And the way lighting design is done right now, without looking at this topic, is like very poor compared to what we can do, knowing the right mechanisms. So I want to close off with a little bit of shameless self-promotion. I didn't get into like the data science things that I do. So here are just two quick shout outs. One, if you look at working with wearable light logger data, I'm making a package to facilitate working with that kind of data. So that's, for example, where the graphic about the light distribution throughout the day came from. And the other one is called LETG Spektran, which is a tool, basically an educational tool on how to work with spectral distributions, see how spectral weighing works, how different action spectra affect or interact with spectra. And this is like a free tool to use. It's completely open source. So yeah, if you're interested in that kind of thing, go ahead. Thank you very much.
Right. Thank you so much, Johannes. That was brilliant and a really nice overview of all the different areas that you can go into when you're doing this kind of research. So thanks again. And so for the attendees, if you have any questions, just put them in the Q&A now, and I will have a look and moderate them. Yeah, before we go there, maybe let me ask a first question, Johannes. So I thought especially your last study was very interesting because it really shows that we're not necessarily measuring the right things, right? So I was just wondering, so I got this right. So when you look at a specific field that you actually, the cutoff that you had there, masking what we wouldn't see basically, that was like a static view, right? So you're sort of assuming, I guess you have to do that in the first instance, you're assuming that you're looking like at the same thing at the same time, without moving your eyes around, without moving your head around, and so on. So of course, that's not kind of what we do in real life, but I think especially for an office environment, it's probably not, it's actually quite realistic because you would be staring at your laptop or something like that for quite a long time. Have you actually done any follow-up studies trying to like understand head movements or like eye movements and taking this into account in that kind of setup as well?
Yeah, so I mean, there's things we have done and things we would like to do. So what we did in the study, and I haven't had time to show it here, was that we looked at what would happen if we also changed like the viewing direction, because obviously this interacts very much with what we see from our lit environment. And this is like an added effect on top of that. What we don't yet know is like how much of that do we see like in real life behavior, where, as you said, people move their head around, and so that's actually part of a study we want to do as part of Melidos, which is the project I'm working on right now, which is collecting wearable data. And what we would like to do is like add 2 sensors to people's glasses, one without a field of view obstruction and the other one with one, so that we get a better sense of how large is this effect like in everyday life. And because obviously this includes the head movements and everything that goes around. In general, this is building off of what we know today. And so what we often have today are vertical measurements of illuminance. And this goes like one step into a more realistic view of the topic. And there are many more things to consider on how to get even better data. But I think it's an important first step.
Yeah, that makes sense. Thank you. Right, so we have a first question in the Q&A by Lena. She's asking, thank you so much for that overview of your research and the impact on health. Just a quick question, is it more favorable for a healthy environment to have more or less light in the human field of view? And how does this light exposure in the human field of view impact the sleep quality?
Yeah, great question. There is like no one size fits all. So it's not like better to have everything inside or as much of it outside as possible. It really depends on what your goal is at a particular time. So during the morning hours or during the day, what you want to have is as much light as possible from within the field of view. So this really means, you know, when you go outside, obviously the lit environment around you is like very bright in every direction. And if you're inside in small rooms, have as much light as possible coming from the walls. And if you're in a large room, usually you can also include the ceiling into that effect. If you are in an evening scenario where you want to reduce the amount of light reaching the eye as much as possible, but you still have to do some visual task like reading, then you really want to place the light source outside your field of view and just concentrate the light, you know, reflected light on your visual task. That is the only thing that comes to the eye. So it depends on your goal for a particular situation and type.
Okay, great, thank you. We have another question by Jamie Sizer. Have you considered adding skull features, for example, the size of brow, to modeling the impact of light measurement on actual light exposure? This can cut out significant amounts of overhead lighting.
So yes and no. So I mean, right now we are just trying to get a very basic understanding about what impact this has. But there actually is a project about modeling, like you know, human anatomy and seeing how this affects the light stimulus a different way. I think like you would need a different approach for measuring this. The approach we took is basically to create a hood. So you have one field of view that you can apply, but there are different methods, for example, where you have spatial resolution in your measurement. And then you can, after the fact, model different kinds of field of view on that and see how that affects the amount of light coming in. So, you know, working with real anatomy, where you also have like more variance, is something that's likely better done with a different approach there, but still very valuable.
Mm-hmm. Great. Right, then I think we don't have any more questions in the Q&A. So I guess then we just, yeah. Or did you want to say something?
Someone raised? No, someone raised the hand.
Ah, yeah, okay. Tony, right. I'm gonna unmute you. Hi, Tony.
Great, thank you.
Yes.
Sorry, I couldn't unmute myself. Thank you for a great presentation. I really appreciate it. My name is Tony. I'm a lighting researcher in the United States. I think I do work similar in nature. I guess like my goal is to do lighting research to influence lighting practice. And I find your measurement error work to be really interesting. And I'm wondering two things. One, do you have published somewhere recommendations for how people like us who, you know, perform measurements in lighting laboratories to estimate these kinds of things can modify luminance meters to reduce this kind of error? And two, do you know any manufacturers who are maybe trying to or interested in building some kind of attachments to their meters to reduce this kind of error? Thanks.
So yeah, great question. Actually, we open sourced the Radiance hoods. So you can just download them and 3D print them to your heart's content. Obviously, in the first step, this only makes sense if you have like the same model of spectrometer, but you know, you can scale the size to the aperture of the spectrometer you're using. So you can just adapt that design. I don't know of any manufacturer that, you know, offers that kind of functionality or feature. Right now, I mean, this is a first step. And what we are seeing is, because the variance is so large between the different projects, I don't think that this will be a simple, you know, correction factor that you can use or apply. Maybe if you have more data, we can find typical scenarios where you can use a correction factor or another. but we are not at that point right now. So my recommendation right now would be to print the radiance hood, do measurements and recommendations, and please publish that. I mean, we do need more data.
Great. Okay. Thanks again, Tony, also for this question. And so, yeah, you're approaching well, it's 9 o'clock here, it's 6 in Munich. So wherever you are, different time zones. Thanks for making it here today, for being here. And thanks again, Johannes, also for your great talk. So let's applaud once again. And and so, yeah, thank you, everyone. And I guess I see you next week, hopefully, again, same time, same place. And with this, have a great evening, great afternoon, great morning, wherever you are. Bye-bye.
Thank you very much, everyone. Bye-bye.